So let's talk about the structure dictates function theme in biology. This is what I often refer to as the most visible of the biological themes. So let's look into this one. It's a pretty straightforward theme. It's just this concept that the shape of something is going to be very specific to what that something is doing. I always think of this as the idea that if I do my job properly as a biology teacher, I can convert all of the major components that we are looking at, all of the major things that we are doing in biology, to really nothing more than interactions of different puzzle pieces. The way enzymes work, the way your immune system functions, the reason why a mitochondria looks the way it does is all because of the function of that process. And the process basically selecting only the best versions of those different puzzle pieces, you eventually end up getting a very, very specific system that is derived from the structure that is coming directly from how the thing functions. And so, be a couple examples here because there's really not a lot of nitty gritty, like individual details in this theme. It's really more in the application of the process. When you look at something like an enzyme, so enzymes are how we digest things. Enzymes are basically what we talk about when we talk about different vitamins working inside of your body. We use enzymes to build things. We use enzymes to break things down. And so when we're talking about enzyme action, really what an enzyme is, it's a protein that has a active site. And so think of that as the puzzle piece where the two pieces come together, right? And this active site is going to be very, very specific to a substrate or the other puzzle piece that locks into it. And most of these enzymes, they do one thing. For the most part, an enzyme's job is to either make something or break something. And sometimes along the way too, in the making and the breaking, we might activate something as well. And we'll talk about that when we get to signal transduction pathways later on in these videos. But realistically speaking, the reason why the substrate in this picture is binding to that region of the enzyme called the active site is because there's all these different charges, little partial positives and partial negatives that are on that substrate that are going to bind to the opposite charge in that active site. And when those things come together, you're going to get something we call a conformational change. And a conformational change, which, by the way, follows this process that we generally refer to as an induced fit, where the pieces come together like the puzzle, that conformational change, that transduction is going to occur, which will either squish together different parts of that substrate, making a bigger substrate. In case of things like digestion, it might actually break apart some of those covalent bonds that are maybe storing the energy of sugars. And so the reason why you're able to digest certain sugars is because you have an enzyme that has a very, very specific active site that will cause an induced fit on that carbohydrate. And then that induced fit is going to cause a transduction to occur where the shape of the enzyme changes. And a lot of times for something like a sugar, we break covalent bonds that are holding together hydrocarbons. And in doing so, we release the energy that we can use to do things like make pyruvate to feed the mitochondria to get ATP. And really, even when we do something like utilizing an ATP molecule for energy, that is going to require us to break some bonds. And so you have to have enzymes that actually do that, which, by the way, most of those enzymes are called kinases. And so realistically, even though we have hundreds of thousands of different proteins inside of our cells that make all the different stuff work inside of our bodies, each one of those enzymes, each one of those proteins, they are a very specifically shaped puzzle piece that is either going to form or break bonds, and it's going to create or break down things from those pieces. Give you another example. You guys have all heard of mitochondria. I know everyone talks about mitochondria and the powerhouse of the cell and how biology teachers are just obsessed with the mitochondria, and that's for good reason, which we'll talk about when we talk about cellular respiration later on. But you may notice when you see pictures of cells that some of the stuff inside of the cells have very distinctive appearances, right? And so the mitochondria is one of those. I always think of it looks like a little jelly bean with squiggly lines inside of it, right? But realistically, those squiggly lines are membranes, and they're squiggled on purpose because the more you fold a membrane, the more surface area or more area that you get for a lot of these different reactions to occur. Turns out for a mitochondria to function properly, it needs a lot of surface area. And so in order to get that surface area, we fold the membrane back and forth inside of an outer membrane. And there's a little bit more that goes into that, but we'll get to that again later on as well. But just keep in mind that if you take a mitochondria and you take those folds out of the membrane, it will not make ATP as quickly as it would if you had the maximum number of folds. And so the reason why a mitochondria is almost every single one of them almost the exact same size, and all of them have about the exact maximum number of folds inside of that inner membrane, That is all because if you don't have that surface area, you will make less ATP, the process of cellular respiration. And again, we'll get to the details on how cellular respiration works, but the important part here is just to keep in mind that the reason why the mitochondria has the structure that it does is because that structure is very specifically tied to the function of the mitochondria, which is breaking down pyruvate and making ATP for us. The same thing for like the structure of most of our body organs, and probably of all of the organs in your body, one of the craziest ones are our lungs. Our lungs are this crazy delicate organ that are essentially filled with lots and lots and lots of little air spaces. Those air spaces, which we call pulmonary alveoli, are pretty much all the exact same size, and they are stacked so densely inside of your lungs that it actually gives your lungs like a Nerf football structure, almost like a spongy structure. And that's because in order for us to get the maximum amount of oxygen out of the air that we breathe in, and to release the maximum amount of CO2 from the bloodstream into the lungs for us to breathe out, in order for that to happen properly, there is a maximum perfect size to each of those pulmonary alveoli. And so this is one of those situations where the structure of the lungs is perfect to the function, which is going to be maximizing that diffusion rate. Last one to talk about here, you may have seen these things before, but it's just kind of a fun concept when we're talking about organism communication. You may have heard of a pheromone before. Pheromones are little proteins that are released into the air for communication. We actually release them too. Pretty much all organisms do. Even bacteria release an equivalent of a pheromone, little proteins for communication process. And in the case of something like, let's say, a moth, moths can't make phone calls, right? They can't necessarily talk to each other, at least not the way we do. And so in order for, let's say, a female moth to communicate with a male moth, they produce these little proteins that they release into the air. And those proteins float through the air and they get picked up by those long antennae on the moths. You may have heard those antennae are kind of like the moth's nose that it's using to smell different things, which is, kind of the equivalent for us, right? Those little antennae have little puzzle piece structures on them that will fit only just that one specific protein that is released by a moth of the opposite sex, which is then going to communicate maybe the location of the moth or that it's willing to or able to reproduce. And so one moth makes the pheromones, the other one has the receptors, and if everything works out properly, the structure of that pheromone will perfectly fit the receptor protein, which will communicate to the moth locations and things like that. This is actually something we've kind of tricked up in the insect world. So if you have, let's say, something like a gypsy moth that you don't want causing problems in your oak forest. Gypsy moths have this nasty habit of eating all the leaves off of our oak trees, or at least their larvae do. So one of the things that we can actually do to prevent a female or male moth from finding each other is we can actually release a huge amount of these olfactory proteins, right? And those olfactory proteins will then bind to receptor proteins. The puzzle pieces lock together, and the moth thinks it's looking for its female or male counterpart, but realistically, because we're releasing those proteins, the moth cannot find each other. And so you end up getting less moth reproduction, less larvae, and therefore less oak trees dying from the larvae eating all of the leaves. It's just a way that we can kind of mess with the structure dictating the function. But again, keep in mind, the entire communication that takes place, all of the signaling between the moths, it all comes down to a puzzle piece, the structure of that puzzle piece being very, very specific. And even though there might be 1,000 different moths in an area, all release different olfactory proteins, the receptor is going to be specific to that one specific moth, that one specific species, and that communication is only going to affect because that structure is very, very specific.